All right, well, let's get started. Thank you everyone so much for joining this afternoon for the second Technoscience Thursday at 2 webinar. My name is Julie Hinderer. I'm the Science Programs Manager with the Great Lakes Fishery Commission in Ann Arbor, Michigan. If you don't mind, if you want to, drop your a little intro in the chat and let us know where you're joining us from. It's always cool to see where people are coming from. I meant to do that on the first webinar if you were there, so definitely interested in seeing where everyone's coming from. Yeah, I have a little brief intro here before we launch into our presentations that we have lined up for today. So if I can advance, there we go. Hopefully everyone should be comfortable with Zoom by now, but just a few tips. Please do mute your audio if you're not speaking. And if you have questions for our speakers, there's a couple different ways you can ask those. You can either drop those into the chat and we will be monitoring the chat, or you can click on that react button on the bottom taskbar and there's a raise hand function. And then you can, once we call on you to ask a question, you can unmute yourself and ask your question verbally. And finally, just a little bit of background info. If this is the first time you're joining the webinar, encourage you to check out our webpage and watch the recording for the first webinar we had last month. Provided a little bit more context and background on what we're trying to do with this webinar series. But the key thing is we're trying to connect researchers and fishery managers to have a conversation about how we can be better utilizing all these great technologies that are out there to help us with management in the Great Lakes. So this is the second of 6 webinars that we're planning during the first half of 2025. This is kind of a pilot series, so we might keep this going after that. We're not sure yet. But we'll be doing all the webinars on Thursdays at 2, and the recordings from the past webinars are going up on our YouTube page at Lamprey Control. My colleague Ted is going to be dropping some links in chat here for some of these resources. And then on our webpage, you can find the recordings for the past webinars. You can get information on upcoming webinars. And you can also, importantly, sign up for the listserv to make sure that you don't miss any announcements about the future webinars. We don't have the next one scheduled yet. We're skipping March because the Great Lakes Fishery Commission has a bunch of meetings during March, and we want to make sure that our partners in fishery management can attend. So we're skipping March. So the next webinar will be sometime in April. We don't have a date or a topic yet. So make sure you join the listserv so you can hear about that webinar once it's scheduled. So that's all I have. Stop my sharing. So yeah, I'm pleased to introduce our first speakers, both from the Great Lakes Observing System. We have Dr. Shelby Brunner, who is the GLOS Science and Observations Manager, and Dr. Jennifer Boehme, who is the CEO of GLOS. So take it away, Jen and Shelby.
Terrific. Thanks very much, Julie. I'm going to go ahead and get started on these first couple of slides, and then I'm going to hand it off to Shelby. So just to give you a little bit of idea about us, Shelby, if we could go to the next slide. For those of you who are unfamiliar with GLOS, we are the Great Lakes Observing System. We're a nonprofit providing end-to-end data services to support science, policy management, and industry in both the United States and Canada. So whether you're deciding to venture out on a boat or confronting flooding in communities, we want to equip observers with high-quality, up-to-date lake information from the region's observing network. We are part of the Integrated Ocean Observing System, which resides under NOAA, the National Oceanic and Atmospheric Administration. But we are also an independent entity and pursue other projects outside of the NOAA umbrella. GLOS has an obligation to aggregate and share data and information about the Great Lakes as one of the regional associations under IUCE. And one of our goals is to be the primary access point for this type of information in the Great Lakes region and beyond. Next slide. GLOS is fundamentally an organization connected through our network of partnerships data providers, and other relationships. Everything that GLOS does is centered around accessibility, availability, and sharing of data and information for the Great Lakes region. When it comes to data and information, GLOS sits at the heart of the Great Lakes and not just in geography. Next slide. GLOS is moving towards developing an information ecosystem based on a range of priorities. The first of that is facilitating the observing network. We want to grow and sustain the Great Lakes Observing System enterprise. We do that through investing in technology to acquire, distribute, and display information. We also serve in a leadership role to drive observing and information innovation in the Great Lakes. Another priority for us is engaging with stakeholders, connecting the Great Lakes communities to the Great Lakes ecosystem. Our aim is to establish new and strengthen existing partnerships, assess and respond to new GLOS community needs, and empower Great Lakes communities through accessible education, outreach, and research. And finally, we're managing data and delivering information. We're here to support a resilient Great Lakes, advance support for decision-making, whether you're a water manager or you're just trying to figure out if it's okay to go to the beach today. We are aiming to provide the services over a range of different users. Seagull is our primary means of doing that. That's our Marine Internet of Things platform that serves most of our data and information in the Great Lakes. This is a simple, usable, web-based application, and it underpins everything that we do with respect to data and information throughput, portrayal, and sharing. So next slide, please. When we think about the Great Lakes, the vast area and size and the density and volumes of people that are served, it's surprising how little we actually know about them. So whether it's underwater topography, water column properties, biology, benthic habitat, even just shipwrecks, the need to understand them more is of paramount importance, especially when you're starting when the region is starting to experience the impacts of climate change. There's a need for greater coastal resilience, and we're seeing an outsized effect of climate change on underserved communities. Understanding the marine environment, especially as we contrast that to levels of understandings that we have of our surrounding landscape, is going to be critical going forward. It's going to be difficult for us to protect the fragile ecosystem if we don't have a good understanding of it. Next slide. So when GLOS looked at how we could better support the overall health of the ecosystem, we put it in context of data to information transformation. We start with platforms, assets, and sensors that are deployed. The data is observed and collected and transmitted to the cloud. And at that point, it's ready for consumption, connection, and data sharing. We have our human-centered design applications like Seagull that help users from a variety of communities to discover data and information. And the last phase of that transformation lifecycle is for those same users to engage with the data and information. They share it with others, pull in new communities as a result, and then inform their own decision making. Our hope is to take users of information to become observers themselves, where they collect their own data and share it community-wide. And at this point, Seagull is our, as said, is the cornerstone for how we're approaching that particular question. At this point, I think I'll turn it over to Shelby so she can walk you through some of the more detailed aspects of Seagull and our other observations and information sharing. Thanks.
Well, thanks, Jen, and hi, everyone. Pleasure to be with you today, and fun diversity of locations everyone's calling in from. So as Jen mentioned, Seagull is meant to take data and really turn it into information. It leverages a technology platform, and it was novel when we did it, and it's been live for almost 3 years. And we're really excited about what we've done with it and the direction that it's going. So I'm going to do what everyone says to maybe not do and try to do a live demo and hope that my internet behaves. So I'm going to go over to my seagull.gloss.org screen. I see Julie nodding, which means it's
Looks good.
going well so far. So if you come to the Seagull homepage, this is free to access for anyone. And you'll notice that there are some great color schemes. This is a model overlay from the Great Lakes Operational Forecast System. And so I'm trying to move these boxes out of your way for you guys. So there's 3 different models that we currently ingest data for, including surface water temperature, surface water currents, and wind speed. So it provides a scale as well as the modeled outputs. You can click on a spot on the map and get the value that they're predicting for that area, as well as forecast it into the future or look back over the last couple of days. But really the heart of what Gloss does is it aggregates near real-time data platforms into one site that can be viewable by the public. So any of the dots that you see are platforms that we have connected. Green dots mean that data has been reported within the last 24 hours. Yellow dots mean we know it's still out in the field and might be reporting but has went offline for some reason. And then a lot of our assets, which are buoy based, are currently recovered because it is winter and things don't survive well in the winter. But with that said, there are some that are out, and I'm going to just walk you through what you might expect to see when you're coming to our site to look at data. So you can click on a platform, it'll tell you its ID and the name, as well as the owner of the platform, and you can view the most recent measurements that were made. If you want to see the trends available over time, you click on the parameter of interest. You can turn on data points if you like and view the last 24 hours, 7 days, or 30 days of data, or a custom view of your choice. If you are really excited about what you see and you want to know more about the data or get the data set, in the top right, there's an option to download data. And you can download a CSV file of the most recent season information. You can download the image of the graph that's on your screen, or you can go to the ERDAP, which is a database that has a lot of configurations available for downloading exactly what you want over specific time periods for the platform. I'll also note that if you happen to have a favorite type of a favorite platform, or sorry, a favorite parameter that you know you always care about, maybe it's wave height or wave period, you can pin it and it will always be at the top of your list. However, in order for this functionality to work, you do need to create a Seagull account. It's free to do it. I'm just going to quickly log into mine and show you some of the other features that are available for free. So I'm going to go ahead and try to pin wave period, and under your profile, when you create that free account, there's a preferences option. You can select the units of measurement that you prefer. So for our Canadian colleagues and others who prefer to use Celsius, you can opt for just Celsius, or you can select everything as metric. There are a lot of different types of parameters that are available. And I'm going to go right to the waves and say I want to pin my mean wave period as being a top parameter that's available. So that means I'll go back to my main screen, and when I click on, decommission shouldn't be there. Sorry, there we go. So when I click on that platform, mean wave period is now at the top because it's one of my pinned or favorited parameters. You can also favorite a buoy or a tower or a location. I'll note we do host watershed data as well. We don't have a lot of it, but we're working on expanding it to USGS sites, and we have the Heidelberg sites on Seagull. So you can mark it as a favorite and then just see what you care about as opposed to seeing the whole gamut of things. And you can also set an alert, which is a pretty neat feature, especially if you're wanting to do sampling or be aware of when conditions might change. So from a parameter page, or sorry, from a buoy or another platform page, you can select the parameter of interest that you might want. So let's say significant wave height, and I want to know when things are really getting high out there. So I want to get told when things are over 10 feet tall, or you can select below threshold and then save. If you're going to be doing a lot of these alert setting, if you're interested in different basins, I would recommend giving it a name like Southern Lake Michigan SIG wave height, because this is what the text or the email will tell you when it comes in. So when you go to your profile, you can see all the different alerts that you have set up and turn them on or turn them off or edit how often you receive them. For example, when I was playing around with this last week, I set a threshold for wind speed way too low. I set it at 18 knots, and I got a text almost every day that wind was over 18 knots, which was great, but I was trying to look for like high wind speeds, and I just underestimated the amount of winds that we had. I'll also note that we did some testing with GLFC and GLaTos and USGS to get one of their first near real-time GLaTos receivers connected to Seagull to work on sharing the data live with GLFC. We got the first one online. It's in Sturgeon Bay, Green Bay, and you can just see when we get hit. So we don't have the fish tag number identified, but you can see how often we got hits over this platform. So depending on where that goes, we are looking forward to expanding that if that's of interest. And then I'll also note that if you're interested in omics data, we have partnered with a group at the University of Michigan to start displaying omics level information on our site. So if you go over to the map overlays and collect omics data, you can see a graph of the community composition that was sampled, as well as the different environmental parameters that were sampled alongside of the environmental DNA that was captured. So a lot of this data is older, but we look forward to having this continually be updated as they get more samples processed and ready to share with us. Okay. So that worked pretty well. I know I'm almost at time, so I will hustle through the last couple of things I wanted to say. One is this is a variety of platforms. So if you have something that you want to connect with us or something that you would like to see connected, please let us know. We support things from meteorological towers to small lightweight buoys to water quality stations. And this is a glider image from Rayon. So we're working on ingesting vehicle information so you can see transient information as well. This is all stuff that was a fail-safe in case my slides didn't work, and it did. And I'll note that we are constantly looking for new ways for data to be accessible and shared with people, especially as Seagull continues to be developed. It's not static by any means. We're constantly making improvements and evolving it. We have a feature called Nested Seagull, which it's fitting that Rayon is presenting after us because we worked really closely with Rayon on doing our first Nested Seagull with them, which basically brands Seagull with their logos, provides special customizations that highlight their assets for their users specifically and can be seen as a data management source. We are exploring expanding this work and understanding more about Indigenous data management and how we can help serve that need, recognizing the CARE and OCAP principles. We've been involved with the Indigenous Great Lakes Network through a mini grant program as well as involvement after the fact. And we have a proposal that we're working on that was funded, a grant, I should say, sorry, a grant to expand this work in the data management for Indigenous and First Nation communities. So we look forward to doing more of that. And then really quickly, I wanted to share where the observing direction is going at GLOS, because there's something, I have an ask for those of you on the call who are in the region and users of data. So we're looking at co-design, making sure that observations that we put out are fit for purpose and that there's someone who is waiting to use the data or aware that it exists and has application for it beyond just yes, no, is it safe? That's important, but also what else can we do with the data to make sure it's being leveraged to its full capacity? We're also looking to leverage what currently exists out there to try to maximize resources and be efficient with the dollars that we have. And on that vein, communicate what is new, communicate new assets so people are aware and can use them to their fuller extent. And another big one for us is year-round observations, because we do have sort of a black box in the winter because things come out of the water. GLOS is currently undergoing strategic planning, and these are some of the areas that we're going to be expanding into, as well as continuing to work in that we've done in the last couple of years. But to get to what I wanted to ask the community and not cut too much into Rayon's time is there's been a couple of projects in recent years that have augmented existing glatos moorings with additional sensors. So Tim Johnson, Matthew Wiles, and Riley Robinson augmented some Lake Ontario glatos moorings with temperature and light loggers in a profile setting at a variety of locations across the lake to look at overturn dynamics. And they're working on a couple of publications on that information that's been really fruitful. I also am aware of Lake Erie doing oxygen augmentation on glatos moorings. Richard Krause and colleagues published this great paper about lake whitefish dynamics across Lake Erie, looking at effects of oxygen concentration or dynamics with fish movements. So GLOS, in the same grant that we have, that Indigenous work, we also are looking to expand this augmentation work for use for fisheries managers. So I am trying to, I'm working with GLATOS, and we want to know what other regions of the Great Lakes would be valuable to have this. It's not real-time data, but it would be year-round data that could inform your decision-making or help you in some way that we can make progress for. So I'm looking for input on where data gaps exist. What do you currently use? What regions would you like to see this in? And anything else you want to talk to me about? This project is just getting started, and I think it has really high potential, and I would love to work with anyone who is, wanting to talk more and help me learn about what's of value to your community. So with that said, here are contact email addresses. My email is not on here, unfortunately, but it's just shelby@gloss.org. And with that, I will say thank you and turn it over to Julie again.
Thank you, Jen and Shelby. Great talk. Yeah, we have a couple minutes. If there are specific questions for Shelby and Jen, or if you have any input on those questions there at the last slide. Seeing any hands or questions in the chat. I have a question. That's okay. You mentioned like you could download data for the season, but do you have historical data? Like if people were going back 10 years or whatever, do you have sort of archives that are available?
We do, yeah. So as long as the platform that you're looking at has been connected with GLOS, you can download that data through the ERD app. So you can go back as long as we have records. We started, I think our longest platforms have been online since 2011-ish. So yeah, if they've been online since then, we have that data for you to download.
Thanks. Other questions? Well, if you think of anything, hopefully Shelby and Jen can stick around until the end of the webinar. We may have a few more minutes at the end.
Sure.
Great, thanks. So with that, I will introduce our next speakers. So we have Dr. Aaron Fisk, who is the Science Director at Rayon, and Professor and Canada Research Chair in Changing Great Lakes Ecosystems at the Great Lakes Institute for Environmental Research, University of Windsor. And your title's super long, Aaron, I don't know. And then it will, will Caitlin and Riley also be presenting?
No, I'm gonna cover it.
Okay, great. Well, take it away, Aaron.
I assume we can see it.
Looks good.
Yeah, thanks, Julie, and to the tech committee for inviting Rayon to come and give a talk. And I hope everybody is doing okay during these dark times. It's important that we stick together and fight for democracy. Anyways, the co-authors you see on this talk are the Rayon headquarters in Windsor. I'm not going to talk much about Rayon right now, but I can at the end if you want. What I'm going to talk about is these simultaneous detections of fish and collection of environmental data in the Great Lakes. And actually, Shelby during her talk started to show some examples of that. I need to acknowledge a whole bunch of people who arguably could have been co-authors on this presentation and will be on some of the publications. These people have either helped with our interpretation of the data or provided logistical support or part of their projects. And there's a whole bunch of funding agencies that have contributed to some of the stuff that I'm going to talk about today. Everybody, I think, would be knowledgeable about the fact that there's been an explosion of research that uses acoustic telemetry to understand fish movements, both in the Great Lakes and the oceans around the world. We all know about GLaDOS and the Great Lakes, and you can see all the receivers across there. But there's also these networks all over the world, including in Australia and in Europe, and then, of course, in Canada. And I was lucky enough to be a part of this. The Ocean Tracking Network provides kind of an overall umbrella to all of these organizations and data storage. And a recent paper that Jordan Matley led showed where we've done acoustic telemetry on this planet at this point. So we've been collecting a lot of fish data, and it's really revolutionized our understanding of fish movement. So I think everybody knows acoustic telemetry, but basically you take these tags and you surgically implant them in fish, and they send out a message that says, I am fish 14, and it might send out pressure and depth data or acceleration, or we even have predation tags. So if the fish gets eaten, it tells you it's been predated. And if there's a receiver out there to hear it, then we're going to record that information. And having that receiver out there is really key. And like I said, we've learned so much about movement patterns, home range. I have that in italics because that's a little bit of a hot button topic. Habitat preference, survival, spawning, site fidelity, all kinds of things. And it's really changed our understanding, and it's helping us be better managers and conservers of these ecosystems. Over the last 4 or 5 years, though, there's been evolving objectives. So we're getting beyond just, wow, isn't it neat that this muskie swam from the Lake St. Clair all the way down to Buffalo and back, and to understand why are they be driven to move, and in particular, linking that to environmental data. And these environmental characteristics, such as dissolved oxygen or temperature or turbidity, linking those to fish detections or fish presence is really one of the new and top objectives of places like the Ocean Tracking Network, as they've put forward their vision into the future. That's right at the top. GLaDOS has talked about it. NOAA's talking about it. A lot of other people are talking about it. We need to understand a little bit more about how we can link these, because as Jen and Shelby just did an excellent job, there's a lot of environmental data out there, and there's a lot more being collected. And as Shelby showed you, some of this is continuous and long-term, and some of it's real-time. So there's real opportunity to try to bring these two things together. And there has been some efforts and ongoing efforts. Sylvia Ivanova, a postdoctoral fellow at Michigan State University and here at the University of Windsor, took a bunch of data in Lake Ontario and looked at a bunch of species to look at what influences where you find them in Lake Ontario. So she took acoustic telemetry data found specific locations, and then linked it to a whole bunch of data. Now, most of that data is static, so things like bathymetric depth or substrate type. Some of it is continuous, but we kind of lack that connection to that environmental data that we're talking about. So how can we bring these two things together? So I'm going to talk about two things, real-time buoys and then autonomous underwater vehicles, or more specifically, Slocum gliders, and how we can do this. So one of the things that we've been working on and other people are working on is these real-time sensory arrays. Now, the ones that Rayon has used in Lake Erie that I'll talk about actually use a similar technology to fish tags and the similar manufacturer Novasea. So we have these central buoys that sit there. A lot of them have weather stations. They're solar powered. They can have other equipment on it, like that black box is a Suna. It's not real time, but it's collecting nitrate data through the air. And then away from that central buoy, we can put out all number of sensors, but we can put out another sensor that has, or another buoy string that has a whole bunch of different sensors like dissolved oxygen or chlorophyll. Those little yellow ones are temperature, all the way to the bottom, DO. And those black sensors actually send an acoustic signal to the central beacon, and it collects that data, stores it, and then also sends it to us real time. So you can go on the Fathom website through Rayon and actually look at these weather stations, much like you see on GLOS. That data also translates directly to GLOS and is available at GLOS as well. Now, this acoustic telemetry signature is just like a fish tag. So it sends a signal that sends the data. Interestingly, Novasea set this up so that these sensors send the information at different frequencies between 51 and 76 to avoid collisions, but specifically, it never sends a 69 kilohertz signal because that's what fish tags use. So these don't get in the way of detections of fish tags. And there's a whole bunch of different sensors that use a whole bunch of different types of technology, and you can set them up to collect data at different time periods. So chlorophyll A blue versus A red, which would give you chlorophyll, blue-green algae, turbidity. There's a bunch of different ones that you can put out and collect data. And since this is a tech presentation, it's not so simple as it's sending it every 10 minutes. All of these instruments collect a bunch of different information. So the reality is that it collects dissolved oxygen about every 3 minutes, 51 seconds, but at the same time, it's also collecting temperature and sending that information. It's telling us about tilt. So if it's on its side, we know there's a problem. You can use that actually to look at current direction as well. And it also sends diagnostics. And so those things are sent at different time periods. And in the end, you get about 1 dissolved oxygen measurement every 10 minutes. Or in the case of chlorophyll, it's every 25 minutes. Different instruments use different amounts of energy to send it. So you have to think about how you want to do that. And we've done stuff like range testing. So when you work on acoustic telemetry in fish, one of the things you want to know is how far away can we actually detect that tag. So we've done quite a bit of work on this. And what we found is these sensors are about like a V16 tag, if you know fish acoustic telemetry, the larger tags. And we had detections up to over 1.5 kilometers away. And as you move the receiver, the sensor away from the central hub, you lose the number of detections that you're actually going to get. Now that little sensor is storing that data, so you're going to get all the data when you pull them up and clean them and use them. But if with regards to real time, you have to think a little bit about how important the data is. And the nice thing is about these systems is if you wanted to set up even 100 DO sensors in one area, they could all be coming to a central buoy and give it to you, that information. Or if there was a dangerous place to put a buoy, you could put a sensor in there and it could communicate to a buoy where it was less dangerous to have it. And then the interesting thing about this is that hydrophone, which I didn't have on the other figure, also picks up fish detections. So at the same time that we're collecting all these environmental sensor data, any fish that swims by is going to give us the data. And that's real time if you want it to be. And like Shelby talked about, you can set this up to let you know when something happens. So if a sturgeon swims by your array, then you're going to find out about it. So what I'm going to show you is just a little bit of data from just west of Point Pelee. This project actually is not about fish detection. It was an NSERC project with a bunch of collaborators to actually study harmful algal blooms, nutrient concentrations, changes in water conditions that affect water utilities. And not to go into a great amount of detail, we tend to take for granted our water utilities. A lot goes into them cleaning and giving us a healthy, clean water that we can use. And if there's a drastic change in the environment, like a drop in temperature or a drop in dissolved oxygen, or a harmful algal bloom, and they don't know it's coming, it can really disrupt that system and cost a lot of money. So what we're hoping to do is develop these early warning systems for these water utilities. But I won't go into that more because this is about fish telemetry. So over the last 4 years, we've had a whole bunch of these buoys out to the west of Point Pelee. You can see where the water intake is for Union Water Supply System, and they provide all the water to the greenhouses in that area. And just FYI, that's the largest concentration of greenhouses in North America, second largest in the world. You might need that food in the future. And so that's where these receivers are. And then, as I said, we had these telemetry receivers so we could collect fish data at the same time. We put these buoys out end of April, May, and we kept them out there as long as we could, sometime in October, varied a little bit by year and by weather. And just to let you know, these things collect a ridiculous amount of data because they're collecting them continuously. So here's some blue-green algae from a number of different sites that you just saw, or dissolved oxygen at the surface and bottom. Sometimes that dissolved oxygen can get very high. It has a little bit to do with how the sensors are set up and oversaturation and stuff, but there's ways to correct that. But if you look at the yellow, that's the bottom DO, so you can see events where dissolved oxygen went low. One quick thing to note, especially when you're using optics, and if you're working in the Western Basin of Lake Erie, biofouling is a significant problem. You have to go out and clean these things regularly or try to use a wiper. So at the bottom, you can see we left a bunch of these receivers out there that we didn't clean, and eventually they become so biofouled because they're using light and optics to look, they don't work anymore. Something to keep a thought on if you're trying to apply these or use these in the Great Lakes. Now, if you're sticking them at the bottom of Perry Sound at 80 meters, you don't need to clean them, right? It all depends on where you were. And I would probably argue at the Western Basin of Lake Erie is probably the worst place for biofouling that you're going to find. And these are some of the things when we pull them up at the end of the year, they're completely covered in dracinid mussels to the point that even if you have a wiper, it's basically a wiper, and then it's just an encrusting of the dracinids around it, which is probably affecting the results anyways. Okay, so on to fish detections. You can see that over this time period, we had 321 unique fish detections, and you can see the species that are here. The NA at the top is mostly walleye, we're pretty sure. But we saw a bunch of different fish over different amounts of time. And then to try to put this in perspective, this is the relationship between bottom temperature and dissolved oxygen in that area. And I want you to understand what's going on here. So we have 4 stations or 4 buoys. We have the 3 years that we looked at them, and it's from July into October. And you can see the orange is the temperature. So at various times during the year, and these are bottom, so mounted at the bottom, you have this influx of colder water. At the same time, generally, when you get that influx of cold water, you're also getting low oxygen water. And so what's happening here is as that water moves from the Central Basin into this area, depending on the wind conditions and stuff, it's bringing that Central Basin deep water that's colder and anoxic, and it sloshes into the basin where we are. And this is what really the Union Water Supply System wants to know, because if they know low dissolved oxygen water is coming in, then they can do something about it and prepare before it comes. But that also allowed us an opportunity to look at fish detections. And we're just getting into this, so I'm not going to present a whole bunch of it, and we have limited amount of time. But over time and over these things, this is Site 4 in 2023. You can see the different fish detections and the temperatures associated with it. Not a lot going on between May and October with temperature, but then you can also see the dissolved oxygen. So this provides an opportunity to say, okay, what fish are present when there's dissolved oxygen? What happens when we have a slush of dissolved oxygen that comes in? And one of the things we're starting to play with is bringing these things together to almost create like two environmental parameter niches for a species. And I don't want anybody to take this anywhere. It's just a preliminary look that Riley and I have been playing with. But the idea here, as you can see in 2023, smallmouth bass seemed to be around a bit more when there was lower dissolved oxygen, warmer temperature. probably relates to the species and their ability to move or wanting to move, and they could have been up top in the water column. But these kinds of combined data that we can get from these buoys provides this opportunity to better understand the ecology of the species. So real quick, buoys provide a fish detection match to environmental data. So we're collecting all that environmental data. The fish is in the same range. It's a link. Instead of trying to find data somewhere else or trying to use a model to estimate what's going on, it can be real time if you want it to be. The reality is just kind of challenges on how you want to combine these data. And I think we're starting to do it in Richard's paper that Shelby highlighted as an excellent example in Scientific Reports. That was a really good paper. But I think as fish ecologists and how we're using this, we have to think about how we want to bring this data together. One of the things we need to do before we do anything with this data is reach out to the tag owners. We know all the people that had tagged fish that ran into our array. We haven't had a chance to upload this data into GLaDOS. We're working with them to try to do it. But it does provide an opportunity to understand these movement with environmental data. And one of the things we need to do is look at all the other receivers that GLaDOS has out in the region and start to see what happens as we can show this pulse of dissolved oxygen, low water coming in, what happens to the fish, right? And then the potential to develop these environmental data niches as we collect more of this data. And one more thought before we leave this is we have all these acoustic telemetry receivers out there. They're deployed and recovered every year. They are sort of buoys of opportunity to put other environmental data out within GLOS. Okay. Let's talk about slocum gliders a little bit. You can see Lydia there right now preparing to use one of those slocum gliders. One of the things we can do is put these acoustic telemetry receivers on the front of them, and you can see those two receivers. One's a 180, one's a 69, so it will pick up fish detections as it's moving around. In this case, you can actually see a sync tag on there that we put on the buoy to move around and collect data. Actually, that's not Lydia, that's Caitlin. And how these buoys work is they use buoyancy. Oh, I can't say that word. Sorry, I'm dyslexic. It's Julie, will you come on and say that word?
Buoyancy.
Buoyancy. Buoyancy-driven gliders where they can pump water out and become positively buoyant, and then it goes up and it uses its fins to go up, and then they can pull water and become negatively buoyant and go down. Ours also have a prop at the back that can be used at times. They carry a battery along, which allows it to communicate, and it drives the instruments that collect data for you. And you can deploy these things anywhere from weeks to months, depending on the conditions and how deep it is. They put these gliders out in the oceans for up to 18 months. We've had a couple of 3-4 week missions in the Great Lakes. I think we can push it further, but we'll see how that goes. The nice thing is you put them out and they collect data, 24/7, all day. Not to get into great detail because I don't have time, that middle sort of science payload bay, you can move different types in and out to have different instruments on it. And then you can also put other instruments like the acoustic telemetry on it. So you can collect multiple types of data at the same time, depending on how you set up your glider. And there's lots of applications. We've used the gliders in Lake Erie for water quality monitoring, telling Ohio DNR a few years ago when the lake actually turned over so they could do the yellow perch surveys. Lots of applications. Largest purchaser of slocum gliders is the U.S. Navy. There are challenges when you put these things out in the Great Lakes. You have to worry about ships, the potential for recreational nets or even commercial nets, boat traffic, those kinds of things can affect it. And you always need to have vessel support. So we've had a couple of times when we've had a leak in the glider, we've had to go out and run and grab it. So first thing, again, range test, and I'll quickly go over this. Lydia Pollock is giving a talk at GLaDOS in a few weeks. And she'll go over this in much greater detail, but we've run some work in Saginaw Bay looking for tagfish and doing some range testing, also in Northern Huron, but I'm going to limit it to just Saginaw Bay. And then last September, we worked in Lake Ontario at Point Peter in 2024. Thing to note here is the older receivers we were using are called RX Live receivers. They're essentially acoustic telemetry receivers that are slightly smaller and cabled that allow us to communicate with to connect to the glider. This last year, we were lucky enough to be able to use a new type of technology that Innovasi has. It's called the mobile RX receiver. It uses beamforming technology. These aren't available commercially yet, either as a standalone detection receiver or that you can buy for your mobile receiver, but we're working with Innovasi, and I'll show you how much better they work. So this is the RX Live, the older receiver, and some work we did. So you can see the path up in the top left. We put out some stationary receivers, and then we ran the glider back and went in different directions and collected a bunch of data. And the tags ping pretty regularly every minute or so that we could collect a lot of data. And we used three types of tag, 180 kilohertz V9, and then V13 that was using high and low power, 69 kilohertz. And you can see the detection probability, which was kind of a little bit disappointing. So for the V13, the 50% detection probability or detection efficiency was only about 400 meters, was less when it was low. We did detect it at times at greater than 3 kilometers. The 180 never broke the 50% detection probability. So we were a little bit disappointed with this because that limitation of detection range and never even getting close to 100% leads to problems if you're going to use the glider. Depending on your question, I remember talking to Good about this and how disappointed he was about it. Now, more recently, Innovasi allowed us to use this RX receiver in Lake Ontario. And again, you can see the mission that we used from start to end, and we had these stationary detection tags, and we used a different set. We only used 69 kilohertz because this new beamforming technology is not available for 180. And we did V16, which is the most powerful tag, down to the lowest power tag, the V8. And you can see that our detection probabilities were actually quite a bit better. Now, the V8, again, never got to a 50% detection efficiency, but the curve was a little bit further spread out. And the 50% detection efficiencies for the V13 and V16 were quite a bit more. Now, you can analyze range data a number of different ways. If you use a GAM using a two-minute model, you get a little bit more information about what's going on. And so there's a couple things that can happen. There's close proximity detection interference, or what we like to call the donut effect. When tags are too close to a receiver, the signal can be too strong. There can be bouncing of the receiver and collision. So sometimes when you're very close and the tag is powerful, it's harder to detect the receiver. That is likely going on here. We're going to look at pings to look at it more. And then there seems to be a bump of greater detection efficiency at 2 kilometers. And these kinds of detection range things are not that uncommon. What's probably happening is between 1 and 2 kilometers, there's something reducing the detection efficiency. So in reality, if that wasn't there, it would be more of a curvilinear relationship that you want to see. But these are the kind of things that we're going to get into and we're going to analyze moving forward to better understand that. So putting this all together, this new beamforming RX Live receiver gives us much better detection probability. We're still quite a bit below that 0.6, which is disappointing, but Lydia will talk a bit more about this at GLaDOS. Could have to do whether or not you're approaching the tag with the glider or moving away from the glider. Obviously, or from the tag as the glider moves away, the receivers are at the front, so the glider is going to act like a shield a bit. We also use an altimeter on the way down that could be influencing the detection probabilities, and also too the orientation of the glider as it's going down. And then there's other things like thermocline, which we know can influence detection efficiency. All of this will be part of our greater analysis as we move forward. Now, just a couple quick examples where we actually went out and looked for fish. So this is a Cisco study that Todd Hayden left, where they took hatchery arrays, Cisco tagged them and released them into the static array of bright blue receivers. And then we went out with the glider, went along the array and went out to different places. And we found these tag fish outside of the receivers at different distances. So it gave us some unique information. At that time, the receivers were not as full as they are now. We also found a couple of lake trout that were not a part of that project, including the lake trout on the bottom was tagged in 2010, and we were apparently surprised that the tag was still even active. So it gives you an opportunity to go to places where there's not receivers. This Lake Ontario project, which is Tim Johnson. So Tim's been tagging whitefish in Lake Ontario at Point Peter, where that little box is, and a bunch of his fish tags just disappeared. So they released them, and they never really saw them again. And Tim was convinced, and he was largely right, that the fish were moving to a particular area where there wasn't receivers. And he was thinking maybe they died because they didn't show up anywhere else. And so that's one of the things that glider was doing in addition to the range test. So this figure at the bottom, which Lydia and Riley put together this morning, show you these missing whitefish tags that we detected in this array. And we think that most of these are dead fish. So it provided an opportunity for us to figure out where these tags were and what had happened to them. And that's informing Tim's work on whitefish, which is really kind of cool. And then some work we're doing down in Oswego, looking at hatchery rays, bloater and lake trout that we've released into the static array. Again, we've run the glider around the outside of it to see if we could pick out our fish as they leave the array, and we have both the bloater and the lake trout. And you can see those are the purple ones, or the little lake trout hatchery raised and bloater seem to be moving towards the northeast. The green ones are predators from other projects. So we were also picking up these other predators, which was kind of interesting and gave us some more information beyond the array that we had. So gliders and moving forward, they really, we're going to look at the range when approaching and leaving tags, when we're descending and ascending. We're going to start linking it to the environmental data. Glider environmental data is not as easy as loading it as it is off of other instruments and stuff. So we're working through that right now. We're going to look at bathymetry. And again, like I said, Lydia has a talk on Tuesday, February 25th. Really interested in looking at ping rates, potential signal strength. So something that a lot of people have been thinking about for a long time in acoustic telemetry is when you detect that tag, there is a measure of how strong the tag is, how strong the signal is, and that gives you some indication how far away the tag is. When a receiver detects a fish, it could be right beside the receiver, it could be a kilometer away. This might inform us a little bit more. Again, we're going to be doing random or tandem range tests for ARCs Live and mobile ARCs. We're going to put both of them on a glider. We have some Lake Ontario alewife work that's going to go on. And again, we're going to do the same thing we did in Oswego. The other hope is to link these fish detections with the environmental data that we saw and doing more of that as we move forward. And then at the bottom is just some ideas about how we might use this technology and ideas for using it moving forward. And then real quick, because I know I'm probably running out of time and people have other things to do. There's other things that the glider can do. This is a project with the University of Vermont. Bianca is a postdoc working with Jason Stockwell, and they hired Rayon. If you look at the glider in the middle, you see that big yellow part. That's a hydroacoustic bay. And we sent this out for 8 or 9 days in Lake Champlain, and this is some of the data that you can get. So we can also do hydroacoustic work with the gliders. And the interesting thing about that is the glider is not a big boat at the surface, potentially disrupting fish. You can send it out and leave it for 8 days, and as long as it doesn't get a leak, you can potentially leave it for longer. Another potential application for fish work in the Great Lakes. Anyways, I'll leave it there because I've used up my time. Thank you.
Thanks, Aaron. Great talk. Yeah, so just open it up. Does anybody have questions for Aaron or questions for Shelby and Jennifer on their presentation? Feel free to drop questions in the chat or raise your hand.
Can I just ask?
Go ahead.
Hi, Aaron. Nice talk. Have you guys compared the detection efficiency with stationary receivers for both types of sort of mobile?
Yeah, we're starting to do that. And that one picture you saw, there was a range test tag that looked something like this that we actually put on the glider itself. So that also will give us some information. We do have other receivers out there, giving us detection range of the tags themselves, so that we're going to compare to the glider. Yeah.
Okay, because I was wondering whether likely it would be worse than stationary receivers, but the reason was that because of these receivers themselves, because different, or because of the vehicle, right? It's on a moving vehicle, potentially with movement or other noises or shattering or blockage by the body.
Yep, exactly. Yep.
Thank you.
Thanks, Shibo. Any other questions? I see one in the chat here. What would the ballpark cost be to equip a buoy system like the one you presented, except without the fish tag detection capability?
So if you bought the system through Novasi, I'm not going to hold anybody at any prices, and prices are rapidly changing right now. Um, but I think if it depends on so if you just wanted the central buoy with the solar panel and a hydrophone that was going to listen for fish tags, Caitlin should probably answer this, but you're probably looking in the neighborhood of about 50 to 60,000 US or Canadian, maybe 65,000. As you add receiver or other sensors and stuff, that price would obviously go up. And then other things to consider too on the cost of that is cellular, especially in Canada, can be quite expensive. So the first year we had them out, we were real timing everything, and that got quite expensive. You have to think about that a little bit as well. But if you want to reach out and really the person to talk to is Caitlin Johnson, or you could reach out to Novasi about the cost of those things.
Thanks. Aaron, that reminds me, correct me if I'm wrong, but Rayon has equipment that folks can borrow? Can you tell us more about
Yeah,
that?
good point. So Rayon, I mean, I could share my screen and talk about it because I had a couple slides at the end.
Sure.
So Rayon is a CFI funded network. We're now part of Global Water Futures Observatory. So we're funded till 2029. We provide instruments for rent or lease to support research projects, very reasonable prices. We provide expertise in deploying them and helping you with downloading data and uploading data to GLOS. So if you're looking to use equipment or you need some equipment, not just fish telemetry, we have all kinds of RBR and SUNAs and all kinds of stuff. Go to the Rayon website. It's rayon.org. And there's actually a link you can look to our equipment, and you can request to use that equipment. We basically charge about 5% of the value of buying the equipment for you to use for the year to collect your data and stuff like that. I will say all of our buoys are already rented for the next year, but there's a lot of other instruments that are available. Thanks for that, Julie. I appreciate it.
Yeah. Other questions for our presenters?
That was without the fish tech. The fish detection capacity just comes with it. You can't pull it out. Yeah. Each of the sensors is between like $3,000 and $5,000. So like a DO, Caitlin's probably rolling her eyes at me, but the DO sensor is about $2,500. It's self-contained. It'll last about a year for battery and stuff like that. They're pretty robust. They only do dissolved oxygen. They don't do relative RTUs, but and we found them to be quite accurate. We've done a lot of work comparing them to actual YSIs that we put down beside it and water collections that we do. The Novasi sensors are very robust, and they come in the same type of plastic that the acoustic telemetry receivers come in, so they're very robust.
I'll just chime in. This is Caitlin here, but just to say, like the fish detection portion of that is kind of an added bonus. Like it doesn't cost any extra; it just comes with it. So, as Aaron mentioned, our initial intention was not really to look at fish detections; it was to work on that water security project. So it was kind of an added bonus that cost us nothing extra to do.
Thanks, Caitlin. Time for another quick question, if anybody has one. Okay, well, I'm not seeing any, so please join me in thanking our presenters for their time. Excellent presentations. I learned a lot. Hopefully you all did as well. And yeah, make sure to get on our mailing list if you aren't already so that you don't miss the announcement about the next webinar. Thanks so much for joining.
Take care. Thanks, Julie.
Bye, everyone.
Cheers.